everyone. Thank you so much for making the time to join us and hear about some of the fascinating research supported by our organization. I'm Dr. Sarah Hernandez, and I'm the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsor for this webinar, Allianz, as well as all of our donors who have given to the HDF to support the research that we fund. Thank you so much. We have a really exciting webinar today that's focused on cardiac health in HD. And the heart isn't typically an organ that we think of when we talk about brain disorders, but the Huntington gene is expressed in every cell in our body. So understanding what the Huntington gene is doing outside of the brain could have implications for HD treatments. And so it's really research like this that focuses on areas of the body outside of the brain that is forward thinking. We're going to need to know about the effect of HD in other organs if future treatment options will treat the entire disease throughout the entire body. I'm particularly excited to introduce our speakers this month during International Women's Month because they're both women. I'm joined today by Dr. Sophie St-Cyr and Dr. Michelle Gray. Dr. St-Cyr is a postdoctoral fellow in the lab of Dr. Beverly Davidson in the Department of Pathology and Laboratory Medicine at the Children's Hospital of Philadelphia. Her research focuses on the heart pathology in Huntington's disease to improve the quality of life of patients. She's exploring how changes in expression of genetic material contribute to reduced heart size and contractile function in HD. Our second speaker today will be Dr. Michelle Gray. Dr. Gray is an associate professor in neurology at the University of Alabama at Birmingham. While her lab has been primarily focused on how a different type of brain cell called an astrocyte is contributing to Huntington's, she's more recently been interested in the role that the Huntington gene has in heart abnormalities in HD. Thank you both so much for joining us today and for sharing your work with us. I also want to thank our audience for being here. Please feel free to submit your questions in the Q&A box throughout the talk. Our audience is made up of both HD researchers as well as HD families, so please feel free to ask both technical and non-technical questions. We're going to have about 20 minutes at the end for a Q&A with our speakers, so send your questions in as you think of them, and we're going to address as many of those as we can at the end. Thanks again to everyone so much for being here. And we'll get started by passing it over to Sophie.
Hi, everyone. So I'm happy today to share my research about Huntington's disease and cardiac health. As Sarah said, I'm a postdoctoral fellow in Dr. Beverly Davidson's lab here in Philadelphia at the Children's Hospital. And again, I want to point out that we do this research thinking beyond the central nervous system in HD because we think it's really important to think of a comprehensive and complete treatment of the disease, and also to make sure we improve the quality of life of patients, of people living with HD. So first, what we know about heart and HD, what we know from people living with it. So let me put my pointer. So first, we know that cardiovascular conditions are very prevalent in the general population, but it's also the case in HD, where it's the most prevalent comorbidity in the disease. What we also know is that the second cause of mortality in Huntington's disease is due to cardiovascular conditions. So usually this is not very typically reported in very great details and is falling under the umbrella of heart failure and cardiac arrest. But it is something that has been reported several times. It's been observed over the last 40 years in several populations in countries ranging from the US to Australia to Denmark and Poland. And this is just a selection of some of these studies that I'm pointing to. The other thing we know from some observations is that there is a decrease in the heart size in HD that is seen late in the disease. And also that there are instances of arrhythmia or abnormal contractions that have been recorded in people living with HD. What this means is that in order for the heart to contract effectively, it needs to have proper electrical conduction in the different chambers. And this needs to happen in a time and a space that is optimal for the expulsion, for for the blood to be forced out of the different chambers. So this is a key function of the heart that we can detect from the outside with an electrocardiogram. We can detect this electrical activity and get a trace like this. This is a normal heart rhythm, and we can detect some irregular heart rhythm through this technique. And Michelle is going to be talking more about this later, but this has been observed in people living with HD. So as in a lot of circumstances, when we don't know that much about what's happening in the people living with HD, we turn to our very handy animal models. And in this case, I'm going to talk mostly about mouse models that have been super helpful in this particular field. And what we detect is, first, there are instances of arrhythmia that are detected in mouse models that present an HD. So they have an expanded CAG repeat in the Huntington gene. I'm not going to dwell on it because, again, Michelle is going to talk about this. But we're going to focus on the fact that we also detect a decrease in their heart size. So a mouse heart is typically small, as you can see here. And in a control mice, that means a mouse with the same genetic background, but without the expanded CAG repeat. They have normal heart size of about 1 centimeter. Well, in the mice with HD later in the disease, we can detect a smaller heart. And here is how we measure it. So we look at the heart size compared to the animal size. So we can alleviate anything else that can bias our observation. But what we see is that if we look at the heart size for the size of the animal, you can see that the control animals in white have kind of the same heart size throughout their life, while in red, the HD mice with HD have a smaller heart with the disease progression. One of the ways that we can look to understand this decrease in heart size is to look at the size of the different cells within the heart. And the muscle cells in the heart are called cardiomyocytes. And we can stain here, this is a microscopy slide, and we can stain their wall in red and then measure the size of the cells. And what you can see is that in control animals, they have a certain cell size that is similar to the mice with HD in early disease or in young animals. And then later on, we see that the mice with HD have much smaller hearts. The cell sizes is much smaller in the mice with HD than in our control mice. So smaller cardiac cells, which could explain this smaller heart. One other thing that's been reported several times, including by our group, is less efficient contractions being present in those mice models. So normally when the heart is in diastole, when the heart is filling up with blood, the chambers are nice and plump. And then when it's time to force out the blood, the heart is contracting and changing shape when the muscle is contracting. And this can be detected by an echocardiography. And an echocardiography is like an ultrasound of your heart. And what it looks like when there's an efficient contraction is that the size of the chamber here gets smaller when the wall is contracting, when the muscle is contracting. So it makes this kind of bumpy trace that we can see here. On the contrary, in an efficient contraction, this trace is much flatter, as you can see in this instance. And this is what we see in HD mice later in the disease. You can think that these changes are minor and might not affect the health of the animal. In the regular time, it's more difficult to detect. But when it becomes very obvious is under situations of stress. A stressor to the heart is something like hypertension or myocardial infarction, and we can mimic that with drugs. And what happens in this situation is that there's an increased amount of blood getting inside the heart. And so the heart has to cope with this increased amount of blood to be forced out. And the typical or well-adapted response is to grow the muscle so it can accommodate this increase in need. However, sometimes there can be a pathogenic response, which is that instead of growing muscle, like here we can see in this microscopy slide in pink, this increase in muscle instead becomes an increase in collagen. So collagen forms what we call fibrosis. It's a sort of scar tissue, and this tissue is not very elastic, cannot contract as efficiently, and also cannot receive the electrical signals as well. So it's really not optimal. This is a very bad case. It's not nearly as bad in our mouse that we've looked at, but in control animals with stress, what we see is normal hard tissue here in white, but under stress, what we see in our mice with HD is increase in fibrosis. So here, it's those red fibers you can see. So knowing all that, what we need is really understand what is going on to explain this heart pathology. This is always their first step to treat a problem, is to really understand the underlying causes. So for this, I'm using an animal model, a mouse model, and I will compare the heart in early disease and in late disease. And the tool we use is gene expression. There's some other very valuable studies that have been made on this, but we're using the latest technology in RNA sequencing to know exactly what is going on in this left ventricle, the one that is the most submitted to stress through time. So what is going on at the molecular level, which genes are expressed. And I've done this work along with Ashley Robbins, which is a very talented graduate student in my lab, in the lab of Dr. Davidson. And what we found, first, I don't want you to understand all of this, but if we're taking all the genes that are differentially expressed, and we put them in this PCA, what we see is that all the red dots, as you can see, these are the animals with HD, are on the right side. On the left side, you have the control animals. So that means that the most difference is due to the presence or absence of the CAG expanded repeat. And then we can also distinguish them by age, where the most of the triangles are on the bottom and the most of the circles are on top. So we can distinguish these animals basically by the presence or absence of the CAG repeat and their age. Then what we do is that we look specifically at those genes that are differentially expressed. And here, each line is a differentially expressed genes, and each column is an animal. And so what you can see overall in mice with HD is that they have this big block of blue, and these are all genes that are less expressed in the mice with HD compared to the control. So overall, they tend to have genes that are less expressed in HD. Then what we can do is to take all these differentially expressed genes that are very helpful. We can look at them individually for what they do, but we can also use bioinformatic tools to group them by function. And this has been very helpful for us. I don't want you to understand any of these, but it's just what we get as an output. What we get very excited about is which functions are, what are the functions associated with the genes that we detect that are differentially expressed. And so I'm gonna talk about them very broadly into three different functions. There's the first one, which is the muscle itself. So we have some components of the muscle, the cardiomyocytes, that are responsible for the contractions. So some of these building blocks are less expressed in the HD mice, the mice with HD, meaning that maybe, yeah, their muscle is a bit weaker to start with. The second main finding is we find differences in genes that are expressed at these cellular junctions. The cellular junctions are the places between the cells, between our cardiomyocytes, that are responsible to transfer information and transfer also this electrical signal. So it's very important that these cells communicate effectively to have this global efficient contraction. And so what we find is that several genes responsible for a good and tight cellular junction are a bit less expressed in animals with HD. The third finding is regarding what we call channels. So some ion channels, these are responsible for efficient electrical conductance within the heart. So as I said, each cardiomyocyte needs to be able to transfer this electrical signal, and this is done through very tight control of those channels, and several of those ion channels are less expressed in the mice that have HD. So taken together, those changes can be detected in early disease in the muscle, the cellular junction, and in the electrical conductance. This could explain in part what we see in the tissue, but also can explain their increased susceptibility to stress, like all those little changes adding up and making them more susceptible to stress through this impaired response to stress. And so what we can now think while studying more the impact of these genes, we can try to think about leveraging existing molecules, existing treatments to ameliorate the heart function. And I'm happy to present a study that we've published last year. I've done this with also the help of a former colleague in the lab, and Dan Child. And one of the aspects we wanted to study was this increase in fibrosis in the heart of the mice with HD. And so along with Navitor Pharmaceuticals, they had an interesting molecule that they thought could be helpful. And so we treated animals with this small molecule orally once a day for 6 weeks, and we submitted the animals to cardiac stress. What we found out is that first, it looked to be safe in the control animals with stress, and the molecule did not show increased fibrosis. But in our animals with HD, we saw a really good rescue of the fibrosis. So the heart was also contracting more efficiently, had less fibrosis. Animals were in better health, were losing less weight and surviving better. And what's really interesting about this is that a molecule analog to this one is in phase 2 clinical trial right now. So we are moving forward. We are working on this actively. My message for this presentation, what I would really like you to remember is, for us, understanding is the first step. So we're starting to uncover what genes and what different functions are different in the HD heart and how we can act upon them. For that, the animal models are very, very helpful, and we can detect so far gene expression changes and early changes in these animal models. What's interesting for us with detecting early changes is that it gives us a window of opportunity to act upon it before there's very detrimental impacts. And the next steps for us is to further understand the HD heart. Now we have several avenues we can take to continue this, to understand better. And also, as I said, leverage existing treatments, including using small molecules that can be protective. And finally, before I give some space to Michelle, I would like to point out that we think that there's an interaction between the heart itself. So there are probably changes that are specific to the heart in HD, but in conjunction to impacts from the central and peripheral nervous system, and that together, both these systems create some of the effects or the detrimental effects that we can detect. So it's important to think of both the heart and the nervous system when we study this. So I would like to thank, as finishing, the Dr. Beverly Davidson, which is my mentor. She has an amazing team here of scientists, very, very bright people, and it's very inspiring to work with them. I would also like to thank the team at Navitor, with which we tested this small molecule, and our some collaborators that are really helpful to give us access to resources that we might not have on site. And finally, a big thanks to the funding agencies, including the Hereditary Disease Foundation. Thank you.
Hello, everyone. Thank you so much, Sophie, for that great introduction. So I probably will be able to skip 2 or 3 slides at least today. So I'm Michelle Gray. I'm an associate professor in the Department of Neurology. And as Sarah discussed, my lab is really interested in a cell type in the brain called glia. But a few years ago, based on some work that we read in the literature about cardiac dysfunction and Huntington's disease, as well as some studies that I have been performing in my own laboratory that demonstrated that some proteins that I was observing altered in expression in the brain was more highly enriched and highly expressed in the heart. So it made me start thinking about cardiac function and or dysfunction in Huntington's disease and how the mutated Huntington protein could impact that. So indeed, you know, as Sarah told you, Huntington is not only expressed in the nervous system or in the brain, but it's actually expressed throughout the body and in organ systems and an organ like the heart. So when we start to think about cardiac mutant Huntington expression or Huntington expression, we have to be reminded that the heart is not acting in isolation. There's actually this neurocardio axis, right? So as Sophie already described somewhat to you guys, there's this autonomic nervous system innervation, peripheral nervous system innervation into the heart, this parasympathetic and so-called sympathetic nervous systems that help to control the action and activity and heartbeat of the heart. So here I'm showing you as an example, a normal rhythm, a normal heartbeat as demonstrated by an echocardiogram. So this is a demonstration of the electrical impulses that are passing through the chambers of the heart and shows us the heartbeat. Let me see if I can get the pointer here. So shows us the electrical impulses as they pass through the different chambers of the heart. But as Sophie already described to you, mutant Huntington expression is present in the cardiomyocytes in the heart. And these cells are also, as one can imagine, critical to the heart function. And mutant Huntington is expressed here. And I think Sophie made a great conclusion where the expression of mutant Huntington, both in the cardiomyocytes as well as the nervous system that's innervating the heart, are critical to the proper function of the heart and ultimately may contribute to the dysfunction. Mutant Huntington in these cells contribute to the dysfunction that we're seeing in the heart. So one of the early studies that was performed in our field, at least in the Huntington mice that I use, the mutant Huntington expressing mice that I use, the BAC-HD mouse model, was performed by Chris Caldwell's group at UCLA. And so what he showed in his work was that there was a change in the heart rate in the BAC-HD mice. So he measured heart rate variability. So this measures the beat to beat interval and reflects the balance of the peripheral nervous system that we've described, the sympathetic and parasympathetic input to the heart. And so here in open circles, here you see the back HD example of the heart rate, and he's showing this over a 24-hour period. But what I think you can appreciate is that there's an increase in the heart rate early in the day in the back HD mice compared to control mice. And so I was sitting at my desk one day, literally, I think about a week before Christmas, and I was thinking about this observation by Chris Caldwell's group, and I also was thinking about an observation I had made about this protein expression in the brains and this highly enriched protein in the heart. So I started to think about how can I work on this? So I work in the brain, but what I did, I really just searched the database of cardiovascular researchers at UAB, and I came across Stephen Pogwiz. So this is an awesome collaborator. So he actually works on some of those junctions that Sophie was telling you about. And he wrote a book on arrhythmias. So I thought this was the perfect person to perhaps collaborate with and communicate with about my project. So I emailed him, we set up a meeting, and ultimately we set up a collaboration with one of his colleagues, Dr. Sabina Huki, also in the Division of Cardiovascular Disease. So we set out to analyze the back HD mice in a little bit more detail. I'm not going to show you all of that data, but basically, I'm going to show a few snippets of that work. So there are indeed cardiac abnormalities in these mice. So this is a histological stain to look at cell types and observe fibrosis. So sort of this scarring that Sophie alluded to in the tissue of these hearts. And so I think you can appreciate over here in the back HD mice heart compared to the wild type that there's a little bit more purplish, bluish stain here. So this stain indicates an increase in fibrosis, an increase in scarring in this back HD heart. And so the percentage is significantly more in these hearts from these animals compared to wild type. So this area that's actually showing the significant fibrosis is part of the cardiac conduction system. So this is part of the area that's actually innervated by the peripheral nervous system and helps to control the impulses that go through the heart to control the contraction and relaxation of the heart. So I'm showing you pictorially here, heart at the top, and some of these areas that receive this electrical stimulation from the nervous system and help to control the activity throughout the upper chambers and lower chambers of the heart. The electrocardiogram down here depicts what happens when that electrical activity passes through these different chambers and shows us this very stereotypical electrocardiogram wave that depicts the heartbeat. And so classically, you can analyze this electrocardiogram wave to really sort of determine where there are abnormalities in the heartbeat and rhythm during normal heart function. And so we took this approach to look at, to use electrocardiograms in our back HD mice. So here we did surface ECGs on the animals, and here I'm showing you just a depiction of that ECG, an electrocardiogram, and 2 heartbeats shown here. What we observed in the back HD animals was a widening of this area called QRS. So this area really gives us information about the ventricular contraction of the hearts of these mice. And so what we observe is that there was a significant widening of this QRS in the back HD animals. So we were also wondering, this was about at 6 to 8 months of age, we were wondering if this was a progressive phenotype. So we took some of those, a subset of those same mice, and looked at this QRS width from 8 months to 18 months. I think you can appreciate in the dark squares here in the back HD mice that there's a significant widening over time from the back HD mice compared to no significant changes, maybe in one mouse, but very few significant changes, very little significant change in wild type mice. So this widening of this QRS could indicate that there's a conduction delay. So there's a change in the signals that are coming into the heart, suggesting that the nervous system innervation or signals from the nervous system are altered in this animal. So just to summarize and not go into great detail, what we observed in these mutant Huntington expressing mice was indeed that there was some histological changes present in this His bundle. So this is an area where that signal comes in. And we observed that this, the cardiac conduction system was altered. And one of the measures that we saw when we performed ECG on these mice was a supraventricular and ventricular arrhythmias. So here on the left, you see this normal ECG trace. And here on the right, I think you can appreciate just by observation that this trace is different. And this is really depicting this ventricular arrhythmias. So work from Dr. Diana Rose's group had shown that there was abnormal cardiac function in HD, as well as the work described in the literature by Sophie from other groups across the country and the world, that there were cardiac abnormalities or cardiac dysfunction in HD. So in that particular study that Dr. Rosas published using electrocardiogram, so basically 10-second reads of retrospective studies. So in that particular clinical trial study that Dr. Rosas had performed, one of the inclusion criteria for that study was normal ECGs or normal cardiac function. But what she revealed in that study in patients that basically had total functioning capacity scores above 7, so they were somewhat affected, but really fairly normal, that 25% of that population had abnormal rhythms as shown on the ECG. So the specifics of that is not that important at this particular time, but this was somewhat striking to me that 25% of the subjects in this retrospective analysis had abnormal ECGs. So taken together, we'd seen these alterations in our back HD mice on ECG. We were seeing in the literature changes in patients in a retrospective study specifically, but we were really interested in trying to determine whether this was true in prospective analyses of cardiac function in Huntington's disease. So Sabina Huki and I, we actually started this HD rhythm study really to do an observational study of HD patients to see if we just took a group of these patients, could we detect changes in cardiac function, specifically changes on ECG using Holter monitoring in these people with Huntington's disease and controls. So we've taken that further, and I have solicited the help of my colleague in the Department of Neurology, Dr. Marissa Dean, and continued collaboration with Dr. Stephen Pogwiz to really determine whether people with HD have increased incidences of arrhythmias. And so to do so, we were performing 48-hour Holter monitoring in these people. So initially, that ECG study was a very short interval. And so we really wanted to perform longer-term studies of this population. And so we're doing 48-hour monitoring. I've learned from the cardiologists that you sometimes need to look over time and not just at a short snapshot. And so we also want to determine whether the hearts of people with HD display abnormal structure and function. Remember, we had already seen some of this in our mutant Huntington expressing animal models, and Sophie has shown you so eloquently that that is also present in the animal models expressing mutant Huntington that they use in their laboratory. And ultimately, we want to perform echocardiograms on these HD subjects. And so we got the support from the HDF to start this HD rhythm study, or to further this HD rhythm study. And this study is really being performed and led by Anusha Kumaradin in our group. And she's been excellent at pushing forward this research and this project. So our initial goal was to recruit subjects from our once-a-month Huntington's disease clinic, from our Huntington's Disease Center of Excellence clinic. And we will identify potential subjects based on their total functioning capacity score. This score will be defined and identified by our neurologist, Dr. Dean, and also with age. So we wanted to only include people up to the age of 7. because after 70, not 70, because afterwards you start to potentially get more cardiac abnormalities. And so we were hoping to take ECGs at their in-person visits into our clinic. And we wanted to apply Holter monitors. So this will give us reads of this electrocardiogram or electrical activity in the heart about for 48 hours. So we would apply in the clinic, record, and then 48 hours later, the subjects or participants will return their Holter monitors to us via mail for our analyses. But what we've realized is that coming out of COVID, many of the HD subjects or HD patients were really hesitant to still come to clinic. So we observed some loss of potential study participants. And so we really had to go back to the drawing board and redesign our study. So we decided to take the approach that many of those in the cardiology world had started to take, especially for obtaining Holter monitoring of subjects, and do this in a remote way. So we had to modify our IRB protocol and get approval to obtain study participants remotely. So we will make an initial call to HD study, potential HD study participants by our neurology nurse. So the Jenna Smith in our group, and so that we weren't cold calling patients or subjects. So they would get calls from familiar clinicians and researchers. So once the neurology nurse informed them about this potential research opportunity, Anusha, really, not we, Anusha would call them and give them more detail about the study. Then they would, once they consented, or if they consented over the phone, we would send them this questionnaire, and then they would fill it out, and then they would fill out the consent form. We would get it back, we would review it, make sure everything's okay. Then we would send them this Holter monitor, and they would wear it for 2 days and send it back to us via FedEx. Once we got it back, we would analyze the results from this monitor. The cardiologist would check it for accuracy, and we would also upload the data to our Cubios analysis system. So if there were problems observed on this Holter monitor, the patient was then contacted and will be referred for consultation with a cardiologist. So how do we do this? So we actually started to use this updated monitor, this Trident Holter monitor. So this monitor we found was better for our remote patients because it was easy to apply, and it was smaller and more comfortable. And actually what the cardiologist had observed was that patients actually prefer this type of monitor. So it's shown here where the data is obtained through the Holter that's present here. The Holter is applied on the chest. So this is an advantage over the older technology where you had these 5 leads that would be applied to the chest of the patients or subject study participants. And then this Holter, this monitor that's shown here. Now, you do not have to apply with this new Trident monitor all of these leads to the chest of the subject, and you would just tape it as usual. And we find that you get better accuracy from the placement by the patients, because one can imagine this would be tricky. And it's a patch. So we decided to use this particular approach, and we enrolled so far in our study 45 participants, about 17 males, 28 females. And HD subjects, we were delighted to find that we could enroll 32, and we were almost equally balanced for males and females. And so we enrolled 13 controls. Unfortunately, all of those controls to date have been female. So this is one aspect that we're continuing to work on. So far, we've done Holter monitoring on 18 of the HD subjects, and about 10 ECGs have been completed. We still have some of these HD subjects in process. For the controls, about 8 of the Holter monitors have been completed and 5 ECGs. So just to talk a little bit more about these HD subjects, that we've enrolled. So as I mentioned, we wanted subjects that had greater than or equal to TFC scores of 7. And so we have a great distribution of subjects across this disease stage. So I don't want to talk in great detail about the data that we've obtained, because the truth is we're still really compiling this data to look at these, the electrocardiograms from the Holter monitors. And there is significant amount of data. And I only show just a small snapshot here of the data that will be obtained or can be obtained from this Holter monitors. And so here you can see some of these terms, this heart rate and this QT, and you will notice that some of those terms are also present on this electrocardiogram example that I'm showing. So all of this information can be obtained from this 48-hour monitor that we sent to the patients, and they applied to their own chest. But what I do want to show is an example of an abnormal rhythm that we observed in some of the HD subjects. So as I said, they undergo Holter monitoring for 48 hours. And so what we observed in 2 out of 18 of the HD subjects were these supraventricular arrhythmias. So this is subject 1 at 2 different time points. What you see in green here is an example of an abnormal rhythm. These are really supraventricular rhythms. So you see here through this, in this 3-beat, as well as down here in a 16-beat interval. And then in a second HD subject, we see an abnormal rhythm as well. So this is very interesting and only present in a small subset of the HD subjects that we've examined so far. But this was also one of the observations that we had made in our analyses of our BAC-HD mouse models. So this is very interesting. So this helps us to do that translational studies from observations made in the animal models, as Sophie has so eloquently described, to now observations that we can go back and see if those things truly exist in HD subjects. So we also perform electrocardiograms in the clinic. And so I'm not going to show data about that today, but just to say that as you observed with the Holter monitors, there's a significant amount of information that we're obtaining from these ECGs that we're performing that we have to further analyze. So we have to continue analyzing this Holter monitors data as well as this ECG data. But one of the things we do, we really want to focus on is this heart rate variability. So I showed you perhaps in the first or second slide that indeed was an alteration that was observed initially in the BAC-HD animals by Chris Caldwell's group. And we want to look at this heart rate variability, which will give us an indication of the nervous system function and or innervation to the heart. So these terms or these letters here, no need to worry about what they all mean, but in our analysis, we will look at these different things that are happening in the cardiac conduction system. So presently we've observed that 2 out of 18 people with HD show alterations in heart rhythms. These alterations were not deemed by the cardiologist to be emergent, so it was not necessary to refer them to the cardiologist at the moment. What we are happy about is that we've successfully recruited people with HD for our study. We know that we have to increase the number of our controls recruited, especially males. Clearly, there's a lot of data that we've obtained here and a lot of data remaining to be analyzed. But I just want to leave you guys with what we think about our study so far and the studies that have been performed in the animal models. And I think Sophie described this well. A lot of what we're talking about today are really translational studies, things that may have perhaps been observed in the animal models, definitely histopathologically, and as Sophie's describing, genetically through RNA-seq experiments. But we believe the observation made in these genetic models are likely also observed in those with HD. I want to highlight this collaborative study that's being performed by me and the clinical colleagues. So it's amazing that I have these awesome clinical colleagues who sat at the table with a basic scientist and who listened to my ideas about what was perhaps happening in people with HD. And we designed studies in the animal model, as well as observational studies to be performed in HD and people with HD, and set out to do those studies to really assess, are there truly cardiac abnormalities that we should be concerned about and that we should be exploring. The other thing I want to highlight is that we've shown that this type of study that we're performing now is very feasible. So we really set out initially to perform this study in clinic. So in our HD Center of Excellence clinic, where HD patients would come for their once a month visit. But what we observed is that we had to think a little bit outside of the box to make sure that this study could be performed. So we now think of this as a sort of clinic to home study. So patients can, people with HD or HD subjects could be identified by our neurologists and our nurses as potential participants for our study. We can contact those people and consent them and really mail this technology to them, have them apply it themselves, and then just send it back to the clinic. So we think this is new technology, expanding what we can do without study participants having to come to the clinic. And so with that, I just want to thank those who've been involved in the study, including Anusha in my lab, Dr. Dean and Dr. Sung, who's another neurologist in our HD Center of Excellence, Jenna Smith, who's the research nurse coordinator, and Dr. Huki, Dr. Zhu and Huki, Dr. Huki, who was in Dr. Huki's lab at that time, and Dr. Pogwiz. So I just want to finally acknowledge Dr. Sabina Huki, who was very instrumental in me starting this work, especially the work that we started in the animal model, and especially for her patience with me while I learned about cardiac function, and with her desire to delve into a community in which she did not work. So with that, I'll stop and we can take questions.
Thank you both so much for sharing your work. I love when people can share things that make me think about Huntington's in a different light, and thinking about the consequence of Huntington in the heart certainly does that. So it's really interesting. Thank you both. And thank you to everyone who's been submitting your questions in the Q&A box. Keep submitting those throughout the Q&A session, and we will try to answer as many of those as we can. Remember, they can be both technical, non-technical, just any question you might have for our panelists. Okay, so we'll start. One of the first questions that came in, Sophie, was for you. They were curious, Diane Carlisle was curious what mouse model you were using for your work.
So for my work, we've been using the ZQ175 because they have a slow, like a slower onset and development of the disease. Yeah.
And there was a related question that came in later from an anonymous attendee that said, to what extent are cardiac abnormalities seen across HD rodent models? Do we know?
Pretty much all of them. We have information about the R62 model, the N171. So both transgenic models, full length and fragment, and also knock-in models. Yep. And
then certainly the back HD mice that Michelle had mentioned.
Yep.
So a question we have for you, Michelle, came from Masood Akram. Question for Michelle Gray. What differences have you seen between male and female HD subjects from the Halter data? If there is a difference, then to what extent?
So far in our analysis, so like I said, we've only seen these obvious supraventricular arrhythmias in 2 out of the 18 patients so far, or subjects so far. I have to admit, I would have to ask Anusha if both of those were male or female. But so far, I think one thing that I learned from Dr. Rose's work was that 25% of the subjects in that particular observational trial had concerning ECGs. And so I think, 2 out of the 18 subjects that we've examined had some arrhythmias. So I don't know at present if we will actually observe any differences in male versus female.
Thank you. So this is another question for Sophie. There was some interest in the fibrosis you were showing. So this is kind of 2 people had a question, so it's kind of 2-part. So an anonymous attendee was wondering if the fibrosis could be happening in other organs in the body, or if it's specific to the heart, And then Sean Davidson added to that. He also had that question, and to expand, does cardiac fibrosis have pathologically similar features found in other forms of fibrosis seen in different disease indications, such as the lung or skin? And might targeting fibrosis be more directly an effective therapeutic strategy?
So thank you for the questions. First, we don't have reports, to my knowledge, of other forms of fibrosis really being prevalent in HD. And also the other thing is, for example, the what I've shown towards the end that was helping with cardiac stress. We were doing treatments kind of leading or preceding the onset of fibrosis. So we're not, I think that what we want to prevent is the consequence being the fibrosis, but we treat upstream of that before it impacts. But it's an interesting avenue.
And one thing I would like to add, I feel like collagen comes up in almost every RNA-seq data set we see, right? Like whether it's astrocytes, whether it's neurons. And so even some of these cell types that wouldn't typically use collagen as a substrate. And so I think it's an interesting point, right? Like maybe there's some sort of common pathway that's being affected that's causing this upregulation of collagen. So something interesting to think about for sure. We have a question for Michelle from Addis Mendizabal. Great presentation, she says. Question for Dr. Gray. For the HD participants with subventricular arrhythmias, do you know if they were symptomatic, i.e. chest pain, fast heartbeat? And in general, what is the protocol with cardiology if patients have abnormal EKGs or have symptoms?
So these are all comers, so to speak, from our HD clinic. So these people had not been referred to cardiology by the neurologist. So as far as we know, they had no outwardly presenting symptoms. So as one can imagine, you and I, Sarah, may have actually experienced this. Sometimes we experience what we say, oh, my heart skipped a beat. And so I think some of these symptoms that would probably indicate arrhythmias in people, we don't think of them as being a problem. We perhaps think of them as some of the normal changes that we may, see or experience during our life. Sometimes you drink too much coffee. So all of these, so I think that these particular subjects did not present to the clinic. They did not have some outwardly presenting symptoms. And so the second part of that question was, would they automatically get referred to the cardiologist? So they probably, so the reason we see this is because we were doing this Holter study in these participants. They probably would never complain to the neurologist about this. And if you didn't go look, if you weren't looking in these participants, you may not see it. And so remember, these are also subjects, HD gene mutation carriers that are pretty, quote unquote, normal functioning. So they do not have severe disease at this point. And so I think that they're carrying on activities of daily living. And so they would not just be automatically referred to a cardiologist just from their neurologist, and if they had not undergone any monitoring.
Thank you. So this one could probably, maybe we can start with Sophie, but I think it could be answered by either of you. So an anonymous attendee asked, do you believe the functional and structural cardiac defects in HD mice and humans are CNS mediated or direct mutant Huntington impact on the heart or neurons, basically?
I think Sophie to this. It's a great question. I think Sophie said it best. I think it's both. And so I think that, those of you who know me know I've always been interested in cell autonomous versus non-cell autonomous contribution of the mutant Huntington protein because no cell is in isolation. They are all contacting and communicating with each other. So maybe if I'm sick as one cell, I can impact the function of you, even if you're not sick. But I think what Sophie's work and even some of the work we're doing in the animal models is suggesting that there are cell autonomous, there are changes in the cardiomyocytes that exist. And then some of this human data and even some of the mouse data, especially on the arrhythmias, is identifying changes that may be caused by this nervous system innervation into the heart. So there needs to be some study to separate the two. I think we perhaps can do that in our animal model, but I think it's both. Now, which is the most proximal and the most important? Sophie, I'll leave that to you to
Difficult to say, but yeah, I definitely think there's an interaction, and I think we, as Michelle said, I'm just concurring. I think we should study both aspects and their interaction, just to be on the safe side as well. But yeah, probably contribution from both sides.
Thanks. So this is a question probably for Michelle from Thomas Cintron. Does the impact to the heart start as soon as the HD manifests, or is it delayed? How long once HD manifests can the heart start showing signs of decline?
So I think what's clear from all of those sort of clinical studies that have been published before, and even some of the mouse work, this is there may be small problems in some of the HD gene mutation carriers early, but those problems are not severe. And so in my view, I think, and I showed you one example where we saw that ventricular conduction abnormality at 8 months, we could see it, but it sort of got worse as the animal aged. And so we haven't gone much younger than that. Maybe Sophie's done some experiments in the younger mice, but and the differences in the age of disease presentation of these animals vary. But I think what it suggests to me is that perhaps there are some changes that are happening fairly early to mid stage of disease, and those get worse with age. So I think it takes some time for overt changes to be present in the heart and be seen in HD patients. So I think some of the reasons why we don't see some of these changes overtly in HD patients and or gene carriers is that the disease is slowly progressing. And this is not something we're focused on in Huntington's disease. We're really focused on trying to make sure people can ambulate and walk properly, make sure those with the HD mutation can think well and function in society. And we're not really thinking about what could be happening to their heart on a day-to-day basis. And so I think that's why it's important that we continue to do these types of studies that we're doing, really to understand, does this really affect people's life? Is it really something that we should be doing? So when a patient comes to the HD Center of Excellence clinic, should they also be going over to get some testing by the cardiologist? So I think these are questions that I can't answer as a basic scientist sitting over here in my lab, but I think it's a question for the clinicians in the community to think about.
And I think related to that, right, like related to the day-to-day living, there's an anonymous attendee who asked, can you speculate on what might prevent cardiac dysfunction comorbidity in the majority of the HD population? And I might use that question as a plug for something that people can do right now, they can do today. And it's the same boring stuff that everyone has heard their entire life, but it's eat healthy and exercise, right? There are very few things that we know for sure, without a doubt, that will improve life with HD, and that is one of them. And it's certainly going to also improve your cardiac health. So I think we're at the end of our time. And so I'm going to take this opportunity to thank our speakers again. Thank you both so much for sharing your work with us. It's incredibly lovely. And I think it's really encouraging and hopeful, not only that we're seeing such translation between animal models and patient data, but also that, Michelle, you were able to kind of take something really negative out of the pandemic and use it to improve clinical trial participation. So that's incredibly hopeful. I want to again thank our sponsor, Allianz, as well as everyone who has donated to the HDF. Our audience, thank you so much for listening. And last, but certainly not least, our speakers. Thank you so much, Sophie. Thank you so much, Michelle. I want to let everyone know registration for our April webinar is now open. It's going to feature work from Dr. Osama Al-Dalama, as well as Dr. Sean Davidson, who are going to be telling us about mapping the brain in HD using something called spatial omics, and how that is advancing HD research. So please sign up now, join us next month. And thank you all so much for tuning in today, and we hope to see you next time.